Okay, welcome back. This is our last little video in our discussion on nucleic acid structure, DNA replication, and chromosomes. So we're going to finish up here looking at chromosome structure. So let's get going with that real quick. It's been a nice and short one. So we talked already a little bit about the levels of organization that you see in DNA, and we're going to expand that a little bit and look at some of these levels in more detail. So let's start at the top of the diagram here. Let me grab a pen as usual. Let's take a look at the top here. So if you link these nucleotides together, you get these polymerase chains, and those are the single strands of DNA, and then they are wrapped around one another to form the double helix. So that's the kind of lowest level of organization that we can think about, if you want, is the double helix. And like we talked about before, the DNA is then wound around these proteins called histones to form a structure called the nucleosome. And so that's the next level of structure. And some people call this the beads on a string level of structure because these look like maybe like pearls on a necklace. And so we have these group of proteins called the histones, and that gives us this beads on a string kind of level of structure. And so that's the kind of like second level of structure if we want to think about it. And that is often referred to as the 13 nanometer fiber. And so that's the kind of next level of organization. Is what we then do is we take that 30 nanometer fiber, that basically like linked together histones, those linked together nucleosomes, sorry, and we then loop those. And so we start making these little loops, and those are held together. The loops are held together, so a little bit difficult to see, but this here is a loop of 30 nanometer fiber, and here's another loop of 30 nanometer fiber, those nucleosomes, those beads on a string, and they are all held together by these protein fibers. So the loops can't be kind of released freely, they're anchored together, and so we get all these looping. And so that forms what is called a radial loop. And so these loops of 30 nanometer fibers are called radial loops. You see them in eukaryotic chromosomes, and you see a variation of them in prokaryotic chromosomes. We then can take that structure, which has a diameter of approximately 300 nanometers, so it's another level of compaction or organization. We can then take those radial domains, those radial loops, and we can then loop them again. So here we're looping them again to make a more complex level of structure together. So this is all about compaction. These molecules of DNA in eukaryotic cells are meters long. I think there's something like 3 meters of DNA inside a cell, which may be only 20 to 30 microns in diameter, micrometers in diameter. And so we've got to compact this 3 meters of DNA into the nucleus, which may only be a couple of micrometers, 4 or 5 micrometers in diameter. We've got to get that 3 meters of DNA into this tiny little structure in the nucleus. And so the way we do that is we wrap it to start with around the histone, so that's one level of compaction, and then we loop that structure, and then we take those, and then we loop those again. So you can do this with a rope. If you've got a rope at home, like a climbing rope or a piece of string, even with that, you can take that and wrap it around something, and that means that you're compacting a long linear thing by wrapping it, and then if you take those wrapped histones, if you like, and you loop them, and you anchor the loops in place with some little clips or something, some paper clips maybe, you could then loop those until you get all the way up here to this 700 nanometer fiber, and that's often called heterochromatin. So that's the name we give to this 700 nanometer diameter. So that is now just under a micrometer in diameter, so that's pretty heavily looped up. And then this heterochromatin is basically what we see composing chromosomes which are condensed as in cells which are in mitosis, and so we'll talk about mitosis a little bit later. So if we look at the compaction we get in an actual chromosome, it's about 1.5 micrometers, 1,400, 1,500 nanometers. And so we go from this 2 nanometer diameter structure here all the way up to compacting it into a much more compact structure, the intact chromosome. So let's take a look at the nucleosomes in a little more detail. They're probably the most important level that we can look at in detail. So here are this would be a double helix of DNA. A little hard to see with the red pen, but That's the double helix of DNA being wrapped around these nucleosomes. And so these nucleosomes are made up of these histones. They've got these names here, H2A, H2B, H3, H4. And so then those are held together. The strands of DNA are held together into that nucleosome by another histone called histone H1. And so here is our next nucleosome. And so that's our first level of compaction. So we're dealing with this here, and one of these is being blown up over here on the right. So this is a close-up of this structure over here. So that's a close-up look at the nucleosome. And then if we want to take a look at the kind of beads on a string level, so we're kind of, in terms of where we are in this diagram, so we're kind of looking at this kind of structure here over on the right. You can see that we've just basically got this double helix wound around the histones to form this now chain of nucleosomes into this 30 nanometer fiber structure. So that's just a little bit of a close-up. Then if we take those structures, that chain of nucleosomes, and we look at the radial loop domains, here is a chain of nucleosomes. You've got to bear in mind, this is a chain of nucleosomes. This isn't naked DNA anymore. It's the chain of nucleosomes that's been looped out and then anchored. And so it's anchored in place by these proteins here. And so that gives you this loop of DNA. And so that's like forming this loop, and that's another level of compaction. And then we just take these loops and we re-loop them, and that gives us our heterochromatin 700 nanometer fiber. So the point of this whole discussion is that you can't just cram 2 meters, 3 meters of DNA into a nucleus. You have to organize it by condensing it, wrapping it around proteins, and then looping those, and then looping the loops, if you like, to form structures that we call chromosomes. So that brings us to the end of our discussion on nucleic acid structure, DNA replication, and chromosomes. And I will see you shortly for our next module.